This is the General Genetics Lab project on yeast complementation by Raphael Milangi and Michaela Moran. What is complementation testing? In this experiment, different strains of yeast, which are mutants for certain genes that code for functioning enzymes in a biological pathway, were examined. And complementation testing is the procedure of crossing two mutant organisms, one of which has a known mutation for a particular gene. In this case, the particular gene will be coding for different enzymes in a biological pathway. From the outcomes and phenotypes of the offspring, the unknown mutant's genotype and phenotype can be determined. In the offspring, an allele that may code for a functioning enzyme may have been contributed by one of the parents if the mutant parent didn't display this phenotype. This is referred to as rescuing the gene. The haploid strains of our model organism, we used yeast in this experiment, and they are a simple eukaryotic organism. Usually, yeast will reproduce by budding, which is asexual reproduction, in which there isn't a lot of genetic variation. As you can see, the life cycle of a yeast consists of the formation of an ascus, which gives off ascospores. These ascospores will germinate on either side of the chart. We have the budding period of the haploid phase, where this is just meiotic division. It's asexual reproduction. And also we have actually two types of yeast. We have an alpha type and an A type. When the alpha and the A fuse together, they undergo sexual reproduction. You need both the alpha and the A-type yeast present in order to have sexual reproduction. And once they fuse, they create that diploid organism. And that diploid organism will continue to bud and create more copies. They could create ascus. They could go into the haploid phase, but once you have the fusion of the two mating types, then that's where you get your sexual reproduction in the yeast. Sexual reproduction allows for recombination and random inheritance of genetic makeup for the offspring. The mutant yeast that we used in this experiment include strains that have mutations in certain genes that will code for either a functioning or non-functioning enzyme in this pathway for adenine. Adenine is a nitrogenous base found in nucleic acids. To get to adenine, you need all of these intermediate molecules as well as the corresponding enzymes. We need the starting material, which is seen at the upper left-hand corner, the P-ribosyl PP, and through all of these different intermediates and enzymes marked by the ADEs across the screen, we get to adenine, as long as all those enzymes are functioning. In mutant strands, we have mutated genes, which will be recessive. These will present themselves as non-functioning enzymes in this pathway. The two that we're going to be looking at are the ADE2 and ADE1 enzyme. So our mutant strains are both alpha type and A type. We need both of the mating types of the yeast to do this sexual reproduction. We have a mutant for ADE1, ADE2, and ADE1, ADE2. Now that ADE1, ADE2, that's our double mutant. And this particular strain has a recessive allele for both the ADE1 and ADE2 enzyme in the pathway. We also have the alpha and A types of those mutations. We also have a wild type organism It's one where the dominant phenotype is expressed. So the dominant phenotype in this case will be a functioning enzyme. The hypothesis of all the crosses involved looking at the type of mutant that was being crossed and determining if that gene will be rescued or not. We have here a marking of red and white. A red colony would determine a yeast lacking a dominant allele for a functional enzyme for adenine production. A white colony resulting from the cross would determine that the yeast has all present functioning enzymes. For example, if we took an ADE1 alpha mutant and crossed it with an ADE1 A-type mutant, the offspring are predicted to be red, since the ADE1 mutant is recessive for that ADE1 enzyme, which is essential in that pathway. If it's recessive, that would code for a non-functioning enzyme, and since both parents lack the allele for a dominant, which would code for a functioning enzyme, that would result in colonies that are red and unable to produce adenine. Materials used include YDA plates, sterile loops, an unknown yeast sample. We used alpha yeast number 2 as our unknown, and samples of known mutant yeast of the opposite type. First, the unknown alpha yeast, mutant number 2 sample on YDA plate was obtained, and a second YDA plate was used as the master plate, which was labeled with the crosses performed, the date, and the initials. 3 sections were labeled for the 3 crosses that were performed in the lab, taking the unknown alpha times the A-type ADE1 mutant, the ADE2 mutant, and the ADE1 ADE2 mutant. And these were all given their own separate cell. In the master plate, a sterile loop was used to transfer the unknown alpha yeast to each of the sections. A new sterile loop was used to streak a sample of the A-type ADE1 to the corresponding cell on the YDA master plate. This process was repeated for the other mutants with a clean loop into their respective cells on the YDA master plate. Afterwards, the YDA master plate was placed in an incubator for 2 weeks at 25 degrees Celsius. After the incubation, the YDA master plate was scored, and any changes in color in the yeast offspring were recorded. After the incubation period, it was predicted that the alpha type was an ADE1 mutant, and if it was crossed with an A-type ADE1 mutant, then the phenotype of the cross would be a non-functional enzyme for the adenine pathway, and the offspring would appear red. This prediction was supported by the experimental data. The chart below shows the observations of the different crosses. So we have the unknown type 2 alpha and the AD1 type A, AD2A, and AD1 AD2A mutants, and the resulting offspring for AD1A times our unknown alpha was red. The offspring for the cross between AD2 A type and the unknown alpha were white, and the offspring from the cross of AD1 ADE2 A type times the unknown alpha were red. After the incubation period, the yeast successfully grew to their respective cells on the master plate. When the unknown alpha yeast was crossed with the ADE1 mutant and the ADE2 mutant of the A type, the resulting colonies were red. This may suggest that the yeast in the leftmost and rightmost cells on the plate did not receive the allele for a functioning enzyme for the adenine pathway, specifically the ADE1 enzyme, from either of the parent yeasts, and therefore complementation and rescuing gene in these crosses was possibly unsuccessful. In the chart here, the picture, we have the 3 crosses. In ADE1 A-type times the unknown alpha, that's the leftmost. We again got the red colonies. In the middle, we have the white colonies from the ADE2 cross with the unknown alpha, and in the rightmost, we have the ADE1 ADE2 A-type yeast cross with the unknown alpha. The cross between the unknown alpha number 2 mutant yeast and the A-type ADE2 mutant, the rescuing of an allele that coded for a functioning enzyme may have taken place. The ADE2 mutant lacked a gene for the functioning ADE2 enzyme. The unknown number 2 alpha strain may have contributed an allele for the functioning ADE2 enzyme in the pathway. Because it isn't a double mutant, we can safely assume that the ADE2 mutant is only recessive for that specific enzyme which it is named after. So therefore, it must have been rescued by the genetic material of the other parent in the cross, the unknown alpha strain of yeast. Since we have all the functioning enzymes from where the unknown alpha may have been an ADE1 mutant, since we cross an ADE1 A-type with our unknown, the colonies were red, which may mean that rescuing of the gene for the ADE1 functioning enzyme did not occur in those crosses. It might be possible that the unknown alpha type yeast was an ADE1 mutant. An ADE1 mutant would only have a recessive phenotype for that particular gene. And so when the ADE2 mutant and our unknown alpha were fused, genetic complementation did occur, and the genes for both the ADE1 and ADE2 functioning enzymes were rescued from the opposite parent. This results in a complete pathway to get to adenine synthesis, and therefore the yeast were white from the possible successful rescuing of the gene. From these observations, the unknown alpha strand is most likely an ADE1 mutant strand. While there were no errors made in the study, further experimentation could involve complementation tests between the number 2 alpha type and other mutant strains for the other enzyme alleles in the pathway to investigate more of the number 2 alpha's genotype for these traits. In the study, only ADE2 and ADE1 were the only enzymes experimented with, the complementation tests and possible crossing over, as well as rescuing of the genes. However, in the pathway to get to adenine synthesis, there are multiple other enzymes that must have functioning capabilities from the genetic material of the parents. So further experimentation could involve crossing the unknown number 2 alpha type with an A-type mutant of the ADE5 gene or the ADE8 gene to learn more about the genotype for the unknown number 2 alpha's yeast strand. Thank you.